So, good morning, Professor Blackledge. Thank you very much for your time to speak with us. And I would like to ask several questions regarding your thoughts and ideas about the field of NMR and intrinsically disordered protein. So your work has significantly contributed in advancing of our understanding of intrinsically disordered protein. So what are the most significant changes that you have observed over the last couple of years in this field?
Well, I think since I started working in the intrinsically disordered protein field, which was well over 20 years now, I think the biggest difference has been that there is now a more general acceptation of the importance of this fraction of the proteome in biological function, cellular function, and which was really not the case, I think, 20 years ago. I think it was quite controversial. Structural biology was very much focused on structure, and our understanding of molecular biology didn't really consider this fraction of the disordered proteome. Very often it was chopped off, for example, to determine structures at high resolution, which one can perfectly understand was reasonable within the optic of that process. But I think now it's much more accepted that the disordered proteome is something that we need to understand and is essential for function. That's probably the biggest change. In terms of methodology, I think NMR has always been really important for studying intrinsically disordered proteins at atomic resolution, and will continue to be the method of choice for describing the interactions and behavior of IDPs.
You mentioned that you started almost 20 years ago with this field. So what pushed you towards intrinsically disordered protein and NMR method?
So those are two separate questions. So what pushed me towards NMR actually was 20 years before that. So I trained as a physicist, and in fact I was doing a PhD in particle physics, where you have very little control over the manipulation of the Hamiltonian, if you like, because you're working in a very, very big machine, where the experiment has been proposed a very long time ago by some extremely intelligent people, and you're essentially working in a process to try and make this, try and test these theories. And I was more interested in perturbing my own Hamiltonians and looking to see what was actually going to the response of the experimental system in a more immediate way. And so somebody told me about NMR spectroscopy, and I got very interested in this. And so I changed PhD and started working in NMR spectroscopy. And I've never really looked back. The reason I like NMR, and I think what's attracted me always towards NMR, is we really stand on the shoulders of giants in NMR spectroscopy, because of the level of theory to explain all of the phenomena that we see in an NMR spectrum. Firstly, it's extremely rich. You can see dynamic processes occurring on many, many different timescales about the environment of individual nuclei. And these can be described very precisely by the extremely well-developed level of theory. And that's because of some extremely intelligent people who first of all, discovered nuclear magnetic resonance spectroscopy and then developed the theories associated with everything that we observe. And this means that we can explain potentially everything that we see in our spectrum at a high level of theory. And I think that's something really, it's not unique, but it's really what attracted me towards NMR spectroscopy. Now, intrinsically disordered proteins, I think that because it became quite clear very early on to me that NMR spectroscopy tells you a lot about the behavior of intrinsically disordered proteins, about the dependence of the function of that disordered chain of amino acids on the specific primary sequence. NMR is extremely powerful at doing that, and so that really attracted me. It didn't stop me working on folded proteins as well, but it did catch my attention, I have to say.
Yes, you are mentioning NMR as a very powerful tool for study the intrinsically disordered proteins, the protein dynamics, but it could be very challenging, especially for newcomers. And I would like to ask, what would you say what is the biggest advantage of the NMR for study the IDP, and what should young researchers keep in mind when using these methods?
Yeah, that's a good question. I think if we compare the study of intrinsically disordered proteins to the study of folded proteins, we have, despite the fact that we have been using NMR spectroscopy, and some eminent members of this field who established the use of NMR to study IDPs long before I started, we've been using NMR as a field to study intrinsically disordered proteins for, let's say, 30 years. But we've barely scratched the surface of understanding their very diverse modes of function. And how would I advise? I actually don't think that you need to provide a great deal of motivation to young students, because there is so much to discover. And I think if there's one thing that we can remind young researchers, is that there really is so much to discover. And I think that's really what still motivates me, is that we make discoveries. And that shouldn't be underestimated in experimental science. It's not given to every field to be able to make the kind of discoveries that you can make using NMR concerning the diverse functional modes of intrinsically disordered proteins. And it kind of motivates itself, I think. I don't know, at least it does for me. And it does, I should say, for the people in the lab. I've never had a problem motivating my collaborators. And I've been extremely lucky to work with some extremely talented, brilliant young researchers. So I should say that really, I can't judge the general public, but I can judge my research group, and I've just been very lucky, and they've chosen to work in our lab. But I've never had problems motivating young researchers, because they've always found it very exciting to make these discoveries.
It's amazing that you're mentioning a lot of novelty and discoveries in this field, mm-hmm. right? So, and you are contributing very much in the development of ensembles.
Yes.
And models of ensembles of IDPs. So could you explain why it is important to think about protein as dynamic ensemble and not only like a single structure? Yeah,
because in intrinsically disordered proteins, it's actually true throughout structural biology. Of course, proteins are molecular machines, and without movement, without dynamics, they wouldn't function. This is true of the most rigid enzyme, equally as it's true for an intrinsically disordered protein. Of course, the scale of the dynamics is very different across the spectrum of proteins, and in intrinsically disordered proteins, it's the dynamics that really define their function. And there are different ways of looking at dynamics, of describing dynamics, and NMR is good at all of them, in fact. One of the questions that we need to answer is, what are the different substates of the protein that are sampled by the primary sequence? That, if you like, in more technical terms, we need to define the free energy landscape, the populations, and the differences between these different conformations. That's the first question. NMR is very good at that, because it actually reports on all of them. And so if we can measure enough NMR parameters, we can describe all of these different states. Some of these states will be functional, and therefore, if we can describe all of those states, then we can understand the function and how it transitions from a non-functional state to a functional state, for example. And for that you need an ensemble, because those states are just different substates. The ensemble is just different substates, excuse me. And then the other question is, what timescales do they interconvert? And that's the dynamic aspect. So there's the conformational equilibrium, and the populations, and then there's the timescales and the interconversion rates between these. And of course, that's also very important to understand how it functions, because intermolecular interactions require, to understand that, you need to understand the kinetics between the different substates. And so NMR is also very good at that. And so we need to understand the ensembles, and we need to understand the interconversion rates between the members of the ensembles in the presence and absence of the partner, the physiological partner. And NMR is really, really good at that. And you can do that at atomic resolution. That's the amazing thing. It's really there's hardly anything missing in a protein once you start studying it by NMR spectroscopy that you can't actually observe.
Yes, it's very true. And actually, you are outlining several questions that are connecting to my last question in this interview, because my last question is, looking ahead, what do you think is the most exciting open questions, not only regarding the ensemble, but in general about the intrinsically disordered proteins, and what advice you could give young scientists who wants to contribute to this field?
Yeah, so I think, I don't know if there's one fundamental overriding aspect which needs to be highlighted, but I think there is something which I think is now going to become more and more common, and this is really where NMR has a really important role to play, and that's in integrating the information that we get from intrinsically disordered proteins. And intrinsically disordered domains, these could just be flexible domains, and intrinsic disorder is a very broad definition. Integrating that into reconstituted complexes of biological interest. That means looking at using appropriate methodology to characterize the structure, possibly electron microscopy or X-ray crystallography, even electron tomography, or even, if we're lucky, artificial intelligence, to understand the framework. And then NMR spectroscopy will be able to tell you about the parts that you can neither predict or see using these methods that provide you with that molecular framework. And yet you can see them in an NMR tube. You can reconstitute this. And this is really the next frontier. It's not just a frontier because people are already doing it. But looking at intrinsically disordered proteins in context of their entire functional multi-component complex, that's really important, because NMR allows you to change the conditions. It's a benchtop methodology that allows you to change, if you need to, the salt concentration, the concentration of the partners, the pH, anything that you need to be able to change to be able to see how things are working. You can do that by NMR spectroscopy. You don't have to make another grid. You don't have to make another crystal. You just do it in the same tube.
Yes, that's a very bright future, I think, ahead of us. And with that, I would like to thank you for your time again and sharing your insight about the field that you dedicate in your research career to. And best of luck in your next discoveries.
Thank you
so much
for the invitation. Thank you.